Great. All right, everyone. Well, thank you so much for joining us today for the second of the COVID-19 Research Lightning Talks organized by the COVID Information Commons community. My name is Katie Nam. I am the operations manager for the Northeast Big Data Hub. And this is part of an effort brought to you by the 4 Big Data Innovation Hubs that has brought to you the COVID Information Commons as a web resource, as well as convening a community on these webinars to talk about some of the COVID-related research that has been going on around the country with a group of amazing different PIs and other researchers in combating the pandemic. So we have a very full agenda today with 8 different researchers who are going to be talking about their work. So I won't delay things too much longer, but I'd like to hand off in the beginning to our executive director here in the Northeast Hub, Florence Hudson, who's going to talk to you a little bit more about the COVID Information Commons, what it is, and for those of you who are already familiar, some exciting new updates. So Florence, if you'd like to go ahead.
Thank you, Katie, and thank you so much for leading all these Lightning Talk webinars. You're doing a wonderful job. And thank you very much for all the PIs who are presenting, the researchers presenting, and everyone who's joining today. So as Katie mentioned, the COVID Information Commons is brought to you by the 4 NSF-funded Big Data Innovation Hubs. John McMullen, who's the executive director for the Midwest, so my colleague in the middle of the country, is with us today. And I have to leave a little early because I'm running an ADSA Birds of a Feather session. And so he'll be on with us for the whole day. So we love how we have great teamwork. We also have Meredith Lee, the executive director for the West Hub that's involved, and Renata Rawlings-Goss from the South Hub. And this is brought to you by the NSF Convergence Accelerator Program. If I could have the next chart, please. So we received this COVID Rapid Award for the COVID Information Commons in May as a team, and Columbia were leading it. So we hosted at Columbia University with the IT team, the libraries team worked with us as well. And as you could see, there's a lot of information on this open portal anybody around the planet can use. We have webinar videos, including a user tutorial. Oh, thank you. I love this. We have the Meet the Researchers page, which actually will have the PI Lightning Talks from today. We break them up into the segments and then we put them on that page. And we're also interviewing researchers. And so we may reach out to you for that. And those are there as well. We have global resources, including data sets from over 40 data sets from around the world from 6 different continents. We also have US and international research funding opportunities there, and groups and different events and things that you can look at. On the events page, we have these webinars, other ones that are related to COVID, and then you can always look at the project team, which is basically all our faces, and our PI, Jeanette Wing. So the COVID Info Commons serves as an open resource to explore NSF-funded research addressing the COVID-19 pandemic. It has over 700 awards in it. It has all the COVID Rapids. It has some eagers and SBIRs and STTRs as well. And you can either do a simple search that's customized on the website into the different 8 directorates in NSF for the COVID Rapids, or you can use the Research Explorer tool, which is really fun. And that's a machine learning generated topographical map tool, as well as kind of a finite element modeling view of all the awards, which is really exciting to use. We have a new search mechanism, which is our new news this month, as Katie mentioned. And that includes information provided by the COVID Rapid PIs that we're integrating into the website. So you can look at the award, you could look at the PI, you can look at their ORCID ID, links to their research, collaboration opportunities. And we also are putting the Lightning Talks in there. So Katie will be following up to ask people to fill out the PI survey if they haven't provided information before. After we get this new release out, we're going to start with the 170 PIs that already have given us data. And we're really excited to continue building this very rich data set to enable collaboration around COVID. And so that's all I wanted to share today. And it's wonderful having you here. Thank you so much. And let's think of our ongoing collaboration opportunities to fight this pandemic. Thank you, Katie.
There we go, unmuted. Thank you so much, Florence, for that overview. As she mentions, there's a lot of exciting stuff coming down the pipeline at covidinfocommons.net. Encourage you to explore that resource going forward, and we'll have more updates coming soon on that front. Helen, could you advance the next slide, please? Thank you. So we are going to get into our speaker lineup shortly. Just as a little bit of housekeeping before we have the first speaker kick things off. Given that we have a full agenda today with 8 presenters, we are going to hold Q&A till the end. So kindly, don't unmute and ask any questions during the presentation. You are welcome to share questions in the chat. Our student Haley will be monitoring the questions there and will be relaying the questions to the researchers during the Q&A period at the end. Of course, our researchers are also encouraged to respond in the chat if they'd like to as well, but we're going to hold a few minutes at the end for that period. We also have a thriving Slack community as well that is on our website. You can find it through the contact link at covidinfocommons.net. And actually, Helen, if you could just advance one further. We welcome you to join there. If you are interested, there's a bit.ly link in order if you want to sign up. Follow-up conversation is absolutely encouraged with all of our researchers. It's been very exciting actually to see some of the collaborations getting off the ground between people who have gotten in touch through these events. As Florence says, it's really intended to be a very cohesive community, and there's been a lot of stuff coming out of this from people who are working on similar fields and able to share their data and other resources. I've seen a question about access to the slides, and we're happy to share those following the event. Not to delay any further, I'd like to get the lineup started by introducing Rachel Wu, our first speaker, who is joining us here today from the University of California-Riverside and presenting on her rapid on older adults learning and adaptation. So Rachel, if you'd like to share your slides, we can get started. Thank you.
All right, thank you. So my lab is interested in learning across the lifespan, and we developed a model that looks something like this, a theoretical model where we think all 3 of these things here in the middle are really important for functional independence and cognitive development and things like that across the lifespan. So from infancy all the way to older adulthood. So what we think we need is learning what to learn and how to learn, and motivation to learn, and also the basic abilities to do things like planning or remember things and things like that. And so the idea is that as you're engaging in real world skill learning, you're going to get access to improve all of these things, which then will lead to long-term functional independence in an environment that's changing. And so this is kind of the holy grail for maintaining functional independence in older adulthood, where you're not reliant on somebody else day to day. And so in the model, we kind of conceptualize it as a car. So you're, if you have a lot of cognitive abilities, if your working memory is great, for example, or your attentional shifting abilities are great, then you have, what we call a really robust engine, right? And if you have motivation to learn and adapt, you have the fuel. But the issue is that a lot of cognitive training programs just focus on the engine or just focus on the fuel or something like that, but they don't actually give learners a sense of where to go, right? So for GPS, you would need learning what to learn and how to learn. And all 3 of these things then provide you with the ability to learn new stuff, to adapt in a changing environment. And so this is all this was all kind of fed into this big study that we did that ended up in an NSF Career Award, where we got older adults to take a lot of new classes with us and, and learn a lot of new stuff they didn't want to learn, in the anticipation that there might be a situation in the future where they would have to learn themselves out of a jam, if they didn't want to be dependent on somebody else. And so they were taking painting classes or, like tech classes or Spanish or photography, music composition on an iPad, things like that. So really difficult classes. They did this for 3 months. And so this is a relatively complex slide, but I just wanted to show you real quick. We ran 2 versions of this, and we were in the middle of the 3rd one before COVID hit. But if you see this purple dotted line here, that's kind of where older adults would typically be in terms of cognitive composite scores on this particular assessment we did from the NIH battery. And this is where a control group kind of rested the whole time from pre-test to post-test in the intervention. And then this green group is the first time we ran the intervention. This red group is the second time. And we also did a baseline version before the intervention started. And this is kind of where undergrads would land as well. So this is where middle-aged adults landed. So this gap here is a 30-year gap. So we tried to bridge that gap even in just 6 weeks. So this tells us that there's a lot of promise, and this is probably why that we got the career award, because there's a lot of promise in bridging this cognitive decline, so to say, even just in 6 weeks. So we were in the middle. This is the middle-aged group. Sorry. So we were in the middle of running this, and then COVID hit. Bam! Everything had to change, and we were like, we had to shut down the intervention and everything. And so we turned lemons into lemonade, and so we asked for money to look at how the older adults in our interventions have been doing compared to older adults who are not. Because the entire time in the intervention, we were like, things are going to change. You never know how things are going to change. You're going to have to learn new stuff, and then bam! Everything changes. They have to learn how to do their own nails because they can't go to the nail salon. They have to learn how to fix their own toilets. They have to, learn how to order food online or whatever. And so we were getting a lot of emails, from the participants saying, look at all the stuff I've learned. This is great. And meanwhile, all the older adults that we were talking to who were not in the intervention were telling us about how they were losing social connections and all these things. So we wanted to study this. And so look at how their learning and adaptive behaviors have been changing over time. And their personal beliefs about what they're capable of doing at different ages, and then social connectivity, and then well-being and isolation. And so we actually have a group of younger, middle-aged, and older adults, as well as older who are not in the intervention, as well as older adults who were in the intervention. So we can see like maybe the older adults in our intervention were more similar to younger adults in terms of how they're adapting. And so it'd be interesting to see that. And we can also look at disparities because our sample is approximately 30% racial ethnic minorities, as well as lower SES, lower income participants. And then we can also look at resilience trajectories as well, because maybe if you've dealt with a lot of stress your whole life, this is not actually that much more stressful. So it'd be interesting to tease those things apart as well. So it's a big team of collaborators, and we're really excited to see what comes out of it. Thank you.
Thank you, Rachel. That was definitely inspiring, and so glad your prior research was able to lend itself well to this new effort. So next up, we will have Sara Rampazzi from the University of Michigan speak on her rapid award on using wirelessly powered sensors to scale up verifiable decontamination.
Okay, thank you. You can see my screen. Okay, so yeah, I'm Sara Rampazzi, and I'm working for University of Michigan, but I'm also an incoming professor of University of Florida. And this is the Rapid SaTC grant of wireless technology for quickly verifiable decontamination procedure for N95 mask. This is a joint effort between me and Professor Kevin Fu as part of the consortium N95 Decon. So as you can see, in the past months, we faced several shortage of PPE and masks, and healthcare workers were forced to use these masks with treated COVID patients. And all around the world, we see this shortage. So the CDC has developed different crisis strategies for dealing with the shortage, and one of them was implemented limited reuse of N95 mask after verifiable decontamination procedure. So our research project aimed to protect these frontline healthcare workers by lowering the risk of infection from reuse N95 mask. So we developed reliable wireless sensor technology and procedure for it-based decontamination process, easily deployable, low power, low cost, scalable, and secure to be used in healthcare facility around the world under resources and under N95 mask shortage. But why decontaminate this mask is so hard? Because as the word say, disposable masks are not designed to be used multiple times. So you don't have only to ensure adequate coronavirus deactivation, but you have also to avoid degradation of the filtration performance and also the degradation of the mask sealing and fitting for the wearer, and also avoid cross-contamination of other pathogens like bacteria, for example. So the CDC has identified the moist heat as one of the most easy deployment and most cheaper techniques for decontamination. Why this happened? Because the temperature and humidity required for this decontamination is suitable for heating device already present in hospitals. So these cheap techniques can be used also to be able to reuse this mask up to 5 times. So our project is tackling the challenges to widely deploy this method in hospital. For example, the lack of standard procedure of this type of decontamination, or the lack of specialized equipment. For example, commercially, the heating device can have humidity and not uniform leakage. Each mask requires stable decontamination condition. So it's essential that you constantly monitoring heat and humidity level for each mask. So the current results that we have, and we developed our first prototype. That is this platform that can be used for monitoring heat temperature and humidity level of each mask. And is a wireless system with multiple sensor nodes, one for each mask, with a high temperature resistance sensor and with limited energy consumption. This is a cheap and adaptable consumer device technology that can be implemented on convecting heating system. And everything is monitored by a smartphone app for automated control, and also it can raise an alarm for like aware the nurse in case of a change of environmental condition. So this is the team that is like during this month is working on that. Is a joint effort between like University of Michigan, also University of Florida, and Northwestern University. So we are working to optimize this system. For example, adding energy harvesting on RFID technology to be able to monitoring the things and lowering the power consumption. And our goal is deployment and test this in the clinical settings, and also integrate this system in like common operating procedure in hospitals. And then we will globally disseminate these results via the NSF DECON consortium. So thank you for the attention. This is how my email, so you can like reach me for other information, and this is the website, so you can see all the update on the project, and visit N95 Decon Home for other mask decontamination information. Thank you.
Thank you, Sara, for sharing all of those resources and for your work on this very timely and important practical project. So moving right along, I see that our next speaker is already sharing, and that would be Sara Bowman of the Hauptman Woodward Medical Research Institute. Sara, I'll let you take it away. Thank you so much. Sara, if you're speaking, you're on mute.
Sorry, can you hear me now?
Yes, thank you.
Great. Let's try this again. Okay, so I'm going to tell you a little bit about the work that we're doing at the Crystallization Center, HWI, with our RAPID to enhance high throughput crystallization for structural studies. So we'll actually start with a figure that probably many of you are familiar with from the CDC, where you can definitely see structures of different proteins, like the big red threatening spike protein. And of course, this is one SARS-CoV-2 virus. What we do in structural biology is what we're trying to get at is what do these structures actually look like at a molecular level detail? Because if we can get at that information, we can do better at designing things like vaccines and therapeutics and ways to actually treat what is happening at a very molecular level. So one of the things that's a little tricky is that viruses are really small. And so I just want to give you a framework for thinking about how we look at things like the proteins that are part of the virus. So this is a width of a single human hair, and we can then move down in scale to a grain of pollen, a single red blood cell, an aerosol droplet, all the way down to one SARS-CoV-2 virus. And this is part of why it can be difficult to really probe what the pieces and parts of the virus, the proteins, actually look like. We use structural biology to do that. So this is a SARS-CoV-2 genome. We're really not going to go into this in any detail, but what is really awesome is that we've already done a lot of work as structural biologists in already determining structures of a number of the proteins that exist that are encoded by the viral genome. So essentially, 29 different proteins are encoded. A number of structures have already been solved. And again, the reason why we want to work on this is because these are the red arrows indicate particular targets for possible treatment and vaccine development. So in the crystallization center, the way we do this is actually by doing X-ray crystallography. So in X-ray crystallography, we have a crystal of our sample, we shoot X-rays at it, we get electron density, and we end up being able to get a model of 3D coordinates. One of the actual big bottlenecks there is actually determining the conditions that will generate the crystals. And so that's what we specialize in at the Crystallization Center. We do high throughput crystallization screening trials. So we'd have 1,536 conditions in 1 microassay plate. So this is very efficient, less sample is required. And we have a lot of imaging and robotics and expertise to be able to do this. We've been in operation for 20 years. So our NSF Rapid has really funded this remote mail-in sample from all over the US for crystallization screening experiments for SARS-CoV-2 proteins. So we're really excited about being able to do this. And we've been really working as a resource for structural biology. And what happens is we monitor these wells, each of these wells over time using a lot of different imaging modalities. So one of the things that our Rapid Award actually funded was development of a graphical user interface for all of these crystallization images. So I had an absolutely wonderful student working with me over the past 12 weeks and developing this GUI that incorporates an automatic scoring algorithm that we developed in collaboration with Google Brain and pharma companies and HWI. And so we're literally about to submit this manuscript describing the software, so we're very excited about that. And we've also been doing a lot of successful screening of a large number of SARS-CoV-2 samples. So we've had people from all over the US sending us samples of their proteins, sometimes proteins in complex with a drug, a potential drug, and we are screening those, getting great results with the crystals, and some of those have already generated structures such as this. This is an example of the SARS-CoV-2 M-Pro protease, which is a main drug target. We've actually got a number of structures that are in publication or on bioRxiv already. And so we're really excited about the work that we've been doing. We definitely are interested in, if you have samples that you're interested in screening for protein samples for crystallography, please get in touch. You can check out our website or Twitter handle. And we do have a structural biology channel on the Slack COVID Information Commons. And so please feel free to check that out. Thank you.
Well, excellent timing there. And thank you so much for sharing that information and this resource, which I do hope structural biologists in the audience take advantage of. Thank you. Both Sara and the following researcher have also been profiled on our Northeast Hub website as examples of research being done here in the Northeast. And maybe our student Helen can drop a link to the set of interviews into the chat there for reference. So next up we have Sam Penta from the University at Albany, who's going to talk a little bit about her research on risk perception and information seeking. So Sam, I'll let you go ahead. Sam, if you're speaking, we can't hear you.
Thank you. Thank you, everyone. Again, I'm Samantha Penta. I'm an assistant professor here at the University at Albany. And today I'm going to be talking about our work looking at risk perception, information seeking, and protective actions for COVID-19. This is work I'm doing with colleagues Amber Silver, another assistant professor at the University at Albany, and Lauren Clay, associate professor at DUville College. And I'd also like to take a quick moment to thank the National Science Foundation for supporting this work. Now, the basis of this study is grounded in the notion that how the COVID-19 pandemic unfolds is fundamentally linked to human behavior. what protective actions people do or don't engage in will shape how COVID-19 spreads. So that means that it's incredibly important to understand what people are and are not doing and why they're doing it. But with COVID-19, there's a couple of unique conditions or characteristics that are important to keep in mind. The first is that COVID-19 is invisible, right? You cannot see the coronavirus with the naked eye. So then this begs the question of what cues do people use to understand a threat when the threat itself is not visible? And then how do those cues shape risk perceptions and behaviors, right? The second characteristic that's really important in understanding COVID-19 behavior is the enduring nature of this hazard. Right? The COVID-19 pandemic has already lasted for months, and it's going to be lasting for several more. So then how do these cues change over time? And then how do the associated patterns with risk perception and behavioral responses change over time? So these questions and the study itself are grounded in the protective action decision model, which was developed by Lindell and Perry. And according to this model, things like environmental and the social context, so things like environmental cues, social cues, characteristics of individuals, the information they're receiving and how they're getting that information, that all shapes psychological processes, which include things like risk perception and perceptions of important stakeholders, which in turn ultimately facilitate or lead to behavioral responses, which are themselves also shaped by situational impediments and facilitators. Okay, so we explore this for COVID-19 using a three-pronged data collection approach that involves a survey, collection of news media, and collection of social media data. And I'm going to spend the rest of my time talking about that data collection in more detail. So the main focus of our data collection is on a repeated cross-sectional design survey. This is a web-based survey, and Qualtrics is both administering the survey and generating the sample for us. We're administering the sample in 3 states, Louisiana, New York, and Washington, which allows us to get some geographic comparisons as well as some comparisons with different experiences of the virus itself. We are sampling based on race, gender, income, and age for a quota-based sample. We're using census data to generate those quotas. We are doing 12 waves of data collection. Data collection started in May. The first couple of waves are a little bit longer. So they are about 7 weeks in duration, and they're a little bit larger in terms of our sample size. So we've got 1,500 participants in the first wave, and then a little over 1,300 in the second wave. Starting with wave 3, our waves are exactly 4 weeks in length, and we're aiming for 700 to 900 participants in each wave with a goal of 300 participants from each of our states. We're currently in wave 4 with an eye of ending data collection in the summer of 2021. So we're going to have a year of data on this. The survey content, the survey questions themselves, ask about all those components of the PADM model. And we have some additional questions that look at things like pandemic stress, health literacy, and food availability as well. So pretty comprehensive. Now that survey data collection is being complemented with use of traditional and social media. So the traditional media, news media that we're collecting comes from 2 newspapers from each of our states of focus, and some additional national news coverage provided by CNN Wire, and then we're also pulling from the national coverage from the New York Times. The social media we're collecting is Twitter data from important international, national, and state level actors. So to give you some examples, we're pulling from like the World Health Organization, the Centers of Disease Control and Prevention, and state level public health agencies. Now, our main purpose in pulling this data or pulling these data sources initially was to get more information on those social and environmental cues, but we're collecting this data for the duration that we're running the survey. So these 2 data sets are becoming really interesting and quite powerful in their own right, and offer some really unique opportunities to look at how COVID-19 is being presented and discussed in these 2 media forums. So with that, I'll thank you so much for your time, and thank you to the COVID Information Commons for the opportunity to talk about our work. Thank you.
Thank you so much, Sam, for sharing that. All right, next up we have Murat Kantarcioglu. Probably mispronounced that, my apologies, from the University of Texas at Dallas. So Murat, ready to go whenever you are.
Can you see my screen?
Yes, it's in there, perfect, full screen.
Sorry. Hi, sorry. Good. I think now it's good afternoon for some. My name is Murat. Kantarcioglu. I'm a professor of computer science at the University of Texas at Dallas. This is our rapid work on privacy risk assessment for data sharing during pandemics with Professor Brett Mellon from Vanderbilt Medical School. So as many of you follow the news, as the COVID-19 progresses, there are new questions coming up with respect to whether it's related to race, whether smoking, for example, increases your risk with COVID-19, bad outcomes, etc. So therefore, in order to answer these kind of questions, whether it's a relationship of race and COVID-19 mortality or other factors such as smoking, high blood pressure, clearly we need to collect more data, and of course, this data needs to be analyzed. But unfortunately, or fortunately in some sense, the secrecy and privacy requirements may slow this attempt. If you look at this Science article published in July, you would see that California didn't want to share, California state officials didn't want to share the COVID-19 data details with some researchers, hindering some certain research. So in one sense, they're right, because it's been known that shared data can be easily re-identified. It's been shown in the past many years ago that if you combine different data sets, for example, if you have COVID-19 data that reports zip code, age, and gender, there is a high risk of linking this with voter registration lists and identify the people who are in the COVID-19 patient data. And of course, this has been known for a while, so it's not a new problem. And understanding these risks has been active research over the past 20 years. But still, COVID-19 changed many things. One thing is that now we have, especially with the social media, we have different re-identification sources. This is an example from New York Times, where they profiled many patients who passed away due to COVID-19 in their life stories. So if you go back to this attack scenario, now you may have additional source where you know some people have passed away and their information, and you can use it to remove the targets and rerun re-identification attacks. So therefore, this rapid is really trying to understand the unique challenges due to pandemic data sharing, especially re-identification risks. So one thing we realize is that unlike the past work, the attributes that are needed to be shared, such as smoking or race, can be changing over time. So we need to really understand that. Also, case numbers are changing, which means that some privacy risks may be changing over time. So we need to really update the privacy risks over time as new data sources are being shared with the others. So therefore, this rapidly changing and uncertain behavior of pandemic data sharing impacts the risk assessment frameworks. And for that reason, we are now developing and updating existing models. So our goal is to really look into what's the best utility we can achieve given the data privacy goals, which in this case, to make sure that re-identification risk is low. In other words, it's very hard to re-identify people. And also, what's the best possible privacy we can provide given the data utility goals. And again, our goal is to give as accurate information as possible. The second option comes into play because there is this argument that we should really focus on combating the disease, so data privacy may be secondary. Still, our past research showed that given your goals with respect to data sharing, different alternatives may have different privacy outcomes. So understanding that would be important. Of course, a major direction would be what's the data utility and how to define it, and that's our ongoing research. To conclude, we build our own past work on this analyzing identification risk, and we update and develop techniques based on these unique challenges of time varying and changing information due to pandemic. And soon, hopefully, like in a month or so, we would have a paper on giving general data sharing policies to guide the health officials, like what data can be shared with low risk, what are the higher risk options and its impact. So please follow us if you have any concerns about the data sharing and privacy of pandemic data. So I'll stop here, I guess. I'm a little bit over time. Yeah, thank you.
Thank you very much, Murat. Moving right along, next up we have Ali Rahnavard of George Washington University, who is going to talk to us a little bit about a novel platform for data integration and deep learning on COVID-19. Ali, whenever you're ready.
Thank you so much for the opportunity. So this is a collaboration work at the Computational Biology Institute at George Washington University. A brief, of what we do in my lab is, with the high throughput technologies, we can measure many different omics, and our goal is to see if we can get a better picture using integrating these different omics data, including genomics, metabolomics, proteomics, and viral genomes as well. So in the objective or NSF proposal was to develop a computational platform that includes two different sets, one analytical approaches and softwares to investigate COVID-19 related omics data. So as you've seen earlier in the different presentation, the main source of this pandemic is the virus, and we are focusing on the genome of virus as a resource to investigate how the virus behaves and if there are specific proteins or regions in the genome of the virus that we need to target for vaccine development. So the way we look at the data is using the sequencing data. We get the genome of the virus across all the infected individuals in the pandemic. And we focus on the genome variation, also on the specific region of the genome, including the proteins and other regions like the non-structural proteins. The way we do that, we get these genome sequences for all the individuals in our population. We try to calculate the variation that explain in each of these samples compared to the other samples, and we try to evaluate and see what those variations we see mean. So here I'm showing a roadmap of how we can calculate, how we can look at the genome variations in specific regions. In the first row, what you see is the genome variation correlation to other regions of the genome. And we see 4 regions or 5 here, that they are highly correlated to the variation of the genome that explains across the populations. And 2 of those regions are very interesting. One of them is the spike protein region, which that number you see, 32.5, says that the variation that we see in the genome of the virus across the population is correlated between the S and the whole genome. And also we see for the NSP 3 regions. So the other 3 regions that you look at, they are yellow, they are big regions and they have a subsets. That's why we didn't focus too much on those, because we expected when you take a big region of the genome and you try to look at how that carries the information that the whole genome takes, it should be highly correlated. So we see here that there are specific places in the genome of the virus, like the S protein, the spike protein. We already know that facilitates the virus entrance to the human cells. We see is highly correlated to non-structural protein 3, and those 2 altogether are correlate with the whole genome variation that we see in the population. So as a part of our NSF grant, we developed an approach named OMCLUST. What it does, you give it a set of points. Here the points are the individuals or the strains of the virus, and the 3 when we run it using the information from the 3 different regions. First, the whole genome of the virus. Second, the spike protein, and third, the non-structural protein. We see that those 3, they give us the same communities, and that's consistent across all of them. That suggests these regions are important to be targeted and investigated further. Also, we look at this variation that we calculated across the population to see how those are correlated with the epidemiological data. So we didn't have that much information, except like sex and age are a good example here, that the virus genome doesn't really correlated well with the sex and age as expected. The outcome could, relate, but not the genome of the variation of the genome. So also in our work, what we are doing, part of it is to integrate the omics data that we measure from infected individual, like proteins or metabolites. We want to see how those information, when we collect them together, tell us information that they lead us to hypotheses from the data that we can target them. For example, what you are looking at here, there is an approach we are developing named B-test. What it does, you give it the metabolites information from the patients and the proteins from the patient. And what it tells us, it gives us a block of relationships. What are the metabolites that they are related to the proteins in or infected individuals. Also, we are developing a deep learning approach, which you give it the sequencing data and tells you, in the beginning of when a person gets infected, how the severity is going to look like based on the genome of the virus that we get from the infected person. So as you see, we are developing different, approaches, deep learning approaches to investigate the omics data. The first focus was on the genome, the viral genome, and now we are moving to the metabolized proteins and try to integrate all this information using the deep learning approach. So here there are several students in my lab and also co-PIs from the computational biology institute at George Washington that they collaborate in this work, and it's a team effort. So you can learn more about our approaches and the COVID-19 result. We post them in a web page in my lab website, and also the softwares that we are developing that you can use for investigating your data. With that, I want to thank you for your attention.
Thank you so much, Ali. Really appreciate that. So our next speaker is Jeff Grann of Credential Engine. Jeff, ready to go whenever you are. Thank you.
Okay, great. I'll just share my screen here. I think Ali, we need to stop.
Yeah, I think Ali, you need to.
I just did a stop pod or a stop share. Okay, great. Wonderful.
Thank you.
Okay, great. So hi everyone, my name is Jeff Grann. I work at the nonprofit organization Credential Engine, who's dedicated to advancing credential transparency. I'll just open with saying that the term credential seems like a single thing, but it's actually many different types of things. And for us, a credential refers to all the many different ways that people signal their suitability. That includes traditional diplomas and degrees, but also certificates, licenses, certifications, badges, and more. And we've done our research in that area and identified over 738,000 credentials being offered in the US market alone. So it's an overwhelming number of options for the American people to make sense of. And what we do is we help the public understand those credentials using linked open data strategies and semantic web technologies. And the way it works is that our organization Credential Engine has led the development over the last 3 years of an openly licensed credential transparency description language schema to fully describe credentials in a way that humans and machines can understand. And then on top of that, we've built free technical services to help people use that language on the web. And this issue has really come to the fore with the COVID-19, where people are struggling to understand credentials and use them for responding to both the COVID-19 pandemic specifically, but then the social, societal impacts that have followed from that as people lose long-paying, occupations are no longer viable. So a lot of folks are looking to credentials to help them weather the impacts of COVID-19. And our Rapid Award is an approach of testing new partnerships that can rapidly increase public data about healthcare credentials and healthcare competency frameworks using Credential Engine's registry. So we think this information, if we can make it more publicly available, will help workers transition into the healthcare sector and then help healthcare workers upskill to develop and respond to the fighting the COVID-19 pandemic. So we built this landing page to help onboard our partners. to publish information to the web, and I'll paste a link to that in the chat here to close. The way we've been operating is that we have a history of partnering with state agencies. So think of state workforce agencies or commissions for higher education. These are folks who have often some regulatory approval process for credentials offered in their state. And we now have formal partnerships with 19 different states and regions in the country, all of whom have been engaged to help publish healthcare credential information. For example, here in New York, we partnered with a group called NIATEP on healthcare credentials, and they've kicked off this work as part of our RAPID grant, hosted Zoom webinars with credential providers in their state, and just recently hired a staff member to help facilitate a lot of the back-end data workflows necessary to make this happen at scale. And similar efforts are underway in Washington and Indiana in particular. But we've also been working with quality assurance organizations. This is a national accreditor, the Distance Education Accrediting Commission. They accredit 34 healthcare related institutions, and that impacts, or they have a governance relationship with 690 healthcare related credentials through their members. And we've begun to map their information workflows so that they can begin pilot testing, publishing to the registry this winter with 2 other organizations. We've also worked at the federal level. We've had a partnership with Career One Stop. The Department of Labor supports Career One Stop. It's a website that is charged with assembling data about state occupational licenses and competency models for the public. And we're helping them convert the information that they currently provide via web pages into linked open data strategies using the CTDL schema. And the competency work has kicked off and is already, I think, complete at this point. And then November, we'll be doing the license finder work, which is all the healthcare, well, all licenses in the country, but healthcare licenses in particular. We've also built this widget so that people can make credential information available on their own websites if they'd like to. This is just a basic design, but it can be fully customized using style sheets. And we now have 2,795 healthcare credentials in this widget. So people can find out more information about healthcare careers that way. And really my talk, I wanted to just say that you can help. So please help join this movement. If you're affiliated with a credential issuing organization yourself, you can help by sharing my contact information and the URL with administrators at your campus. It might be a registrar or a provost. That kind of level is usually where we intersect with. Or you might be involved in different quality assurance or regulatory organizations, maybe professional associations, healthcare medical boards. All of those groups have important information for the public about healthcare credentials, and we'd like to make it more publicly available via our services. So I'll just paste the chat in the link, in the chat, and here's my contact information, email as well. Thank you very much.
Thank you, Jeff. All right, well, that leaves us with our last but not least speaker, Praveen Rao, who is of the University of Missouri, Columbia, and we'll talk about his work democratizing gene sequence analysis for COVID-19. So Praveen, whenever you're ready.
Yep. Thank you, Katie. Good afternoon to many of you. It's still 11:50 here, and I'm going to talk about our work on democratizing genome sequence analysis for COVID-19 using Cloud Lab, which is an NSF-funded experimental testbed for cloud computing. I'm at the University of Missouri, Columbia. And so the motivation for our work is pretty straightforward. There's a growing interest in understanding how an individual's genome actually impacts the symptoms that a person sees due to COVID-19, as well as the severity of the disease, as well as the eventual outcome, whether they survive the disease or not. So by doing genomic analysis of the genome of COVID-19 patients, we can improve our understanding of the disease And this can enable us to have new treatment strategies and faster drug discovery. There are a number of publications that are coming up, and one of them is in the New England Journal of Medicine that did a genome-wide association study. And this was about using about 1,900 patients and studying their genetic variants in their genomes. Another effort that has been around is the COVID human genetic effort. And it's an international consortium, and the goal is to basically identify how an individual's genome impacts their response to COVID-19. So our genomes may hold the answers to fight COVID-19, and this is an important area to focus on. So the goals of our project are basically twofold. The first is to enable researchers to perform variant analysis at scale on human genome sequences. And the goal is to give them the resources at no charge. So variant analysis essentially detects variations in the individual's genome, for example, single nucleotide polymorphisms or small inserts and deletes, as well as we can even think of structural variants such as copy number variations. Now, the other part of the research is going to focus on developing an efficient de novo assembly of the human genomes so that we can do deeper analysis of the variants of the genomes of individuals, one belonging to a group that were not impacted by the disease and the other belonging to the group that were impacted by the disease. And in this particular context, we are looking at COVID-19. So to achieve our goals, what we're going to do is to develop a software infrastructure using Cloud Lab. And Cloud Lab has been around for several years. It was originally designed for computer systems research, and it was not really planned for data-intensive workloads. But in this particular effort, we are going to show how we can leverage Cloud Lab and have workarounds around some of the limitations that it has to build an infrastructure that can support large-scale genomic analysis using cluster computing technologies, as well as open source tools. So we are going to be looking at the best practices that are out there for genomic pipelines. One of them is GATK. We're also going to look at the BD Genomics project. We're going to use Apache Spark to achieve parallelism. And we are also going to use some of the open source tools that are widely used in the genomics community. And the second part would be to develop an efficient algorithm that is going to help us perform what we call as exhaustive variant analysis using de novo assembly. So essentially, we are talking about two groups of patients here, and using bipartite graph modeling, we will be looking at the pairwise comparison between these individuals and have deeper analysis of the variants in their genomes that will help us better understand the disease. And on the right side, what you see is essentially the whole ecosystem that we are putting together, leveraging whatever is available in terms of open source software and building our own components, such as the exhaustive variant analysis engine. And the goal is, at the end of the day, researchers should not worry about having to pay high costs for cloud computing resources, either through commercial vendors or other venues. So Cloud Lab is a free academic platform, and we would like to leverage that to basically empower researchers with the ability to do large-scale genome analysis in an effort to find a cure for COVID-19. We also would like to understand how does the genomic workloads impact the computer and network systems. How can we build future systems that are better geared towards processing genomic workloads at scale? Now, here's a project site. We have it actively being hosted on GitHub, and we are able to allow users to sign up on Cloud Lab and do variant analysis on a single node, as well as on a cluster. They can also do de novo assembly on sequences. We have access to two publicly available resources. One is the 1,000 Genomes Project, which is, of course, not related to COVID-19, but it gives us a lot of data to test our software. And then we have access to the COVID-19 data portal, where some of the projects list sequences that are available for us to work with. We also report some performance evaluation on our initial efforts. How fast can we actually do the variant analysis on these sequences in a cluster mode, and as well as de novo analysis. So please follow this link if you're interested in doing large-scale genome analysis at no cost. Here is a simple UI that we are currently building so that you can provide access or provide the URLs for your files, and then you can just say execute and give your email ID, and then we will send you back the variant analysis file once the process is completed. Here is our team. It includes a diverse set of researchers ranging from pathology to genomics to bioinformatics to epidemiology. And my PhD student, Arun Zakaria, is actively involved in building the software along with me. So feel free to reach out to us if you have any questions or interest in using our platform. And thank you so much for your attention.
Thank you very much, Praveen, for sharing that resource. It's been really exciting seeing the diversity of work that is handling all the different aspects of the pandemic. And I know in the chat we've had a very robust Q&A. Haley tells me it looks like all of the questions to date have been answered. If you do have any additional questions for any of our speakers, please feel free to share them there. And a big part of the COVID Info Commons, I think that is something we really encourage and something we've been seeing happening organically is to follow up, to keep having these conversations with our researchers, with each other. I've seen a couple potential collaborations sparking up in the chat already, which is really exciting. And this is some of what we are trying to do here is really help foster this community against this challenge. Haley, would you like to share the new question for Praveen?
Yes, sure. So Miranda Lynch asked Praveen, are there data protection or HIPAA protections in place on the platform?
Yes, so what we do right now is that the users have to click a checkbox that says they are going to use only de-identified data. And Cloud Lab as such has the right security policies to provide a secure environment for computing, but they don't really focus on any healthcare data. So it's our job to make sure that users are only uploading sequences that are de-identified. So that's something is a challenge, but we expect them to agree to a condition, and we rely on their judgment to use our platform for working with genomic sequences. If there are any good suggestions, I'd be happy to take it from Miranda or others.
Thank you, Miranda, and thank you, Praveen, for the answer. And as I said, I do encourage folks to follow up, to continue the conversation, whether directly or leveraging the wider community as part of the Slack and the COVID Info Commons in general. As you can see here, our next event will be held on November 13th from 1 to 2 p.m. Eastern Time. You'll find registration information on our calendar at covidinfocommons.net, as well as a sign up for our mailing list. You'll find the link below. All of that you can navigate to very easily from covidinfocommons.net, and these slides and the talks will be available, of course, online, both on our YouTube channel, on our website, and we'll be sharing it through that newsletter, as well as updates on future events and other resources. So please do sign up if you haven't already. It's been a helpful resource for a lot of folks. Helen, if you could just go to the last slide, we're just going to confirm the last, oops. That might have been, okay, that might be an older version, that's fine. But in any case, we are excited to have our lineup confirmed for the final event, not the final event, for the November event that will be held on Friday, November 13th. That includes Deborah Lafer from New York University, Nima Moshiri of UCSD, Brandon Johnson, who I believe is on the line, of Decision Science Research Institute, Ying Zhang of the University of Southern Florida, Leela Hadayatafar of New England Complex Systems Institute, and Jifeng Yang of University of California, Santa Barbara. Another great group of researchers, and we do encourage you to sign up via this link that Helen is sharing here. There's registration information available there, as well as their full information about their projects. So please do stay in touch. Feel free to reach out to the team at info@covidinfocommons.net should you have any questions, and stay in touch with each other. And in the meantime, thank you to all of our speakers for your great talks today. Thank you to our students and our project team at large for making all of this work possible. And thank you to NSF for their support in getting the COVID Info Commons off the ground. We couldn't do all of this without the whole community. All right, well, hope everyone has a good rest of